Hello, everyone, and welcome to today's keynote webinar titled Live Single Ultra High Throughput for Applications in T-Cell Potency and Small Molecule Drug Testing. This webinar is part of the 7th annual event in the Laboratory Automation Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during this presentation. You can find that live chat located at the left of your screen. You can also participate by submitting as many questions as you'd like during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of this presentation. Now, if you have any trouble seeing or hearing this presentation, just simply click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located at the Abstract tab from the menu at the left of your screen and follow the process to obtain those credits. I now present today's speaker, Dr. Benjamin Yellen, co-founder and CEO at Celldom. For a complete biography of our speakers, please visit the presenter tab from the menu at the left of your screen. Dr. Yellen, welcome. You may now begin your presentation.
All right. Thank you for the kind introduction, and I appreciate the opportunity to be here today to talk about our next generation cell assay that we're developing at Celldom. So we are targeting the cell and gene therapy space, and I think it's an understatement to say that this is a totally new paradigm in medicine. It is just fundamentally different than molecular medicines, which are composed of small molecule drugs and large biologics, because in cell and gene therapy, the cell plays a much more fundamental role in both the manufacture of the medicine, in the case of gene therapies, but also in many cases, the cells are the drugs themselves. And these are typically the most well-recognized applications right now are in CAR T and CAR NK, as well as some B-cell therapies that have recently been FDA approved. And these are the first precision medicines because in many cases, you're using the patient's own cells as the drug that's being engineered and re-injected back into the patient. So this poses challenges that make it just fundamentally different than traditional molecular medicines, because living drugs, which is what these are not only more complex than small molecule drugs, but they evolve in time and they change. And so the standard approach for developing and testing and for purity and potency of small molecule drugs is predominantly mass spec and liquid chromatography, where you can determine and purify these molecules to 99% purity or greater. And then dosing is just based on relative weight of the patient. But when we talk about cell-based medicines, and if you've been around for the last 10 years and seen the rise of single-cell analysis, it's become very clear that no two cells are alike. So the question of whether a sample is pure or not just flies straight out the window. And so we are at a fundamental crux point of how do we actually measure the mode of action and the composition of a drug that's being injected into a patient when they're cells which are all different from one another, and also during the course of treatment, some cells will grow, some cells will be more effective and others won't. So this is the big question to the industry, is that we are in the solution to this is that we need better cell-based assays, next generation cell-based assays to complement these next generation therapeutics. So there's a lot of promise in this field, and there are great challenges as well. So one of the main challenges right now for these precision medicines is just constrained manufacturing. Uh, because you have to individually make a drug for each patient for these cell-based medicines, they're just simply, it's not a very scalable approach, like traditional medicines, which are made in large batches and distributed to patients. And so the problem is that a whole different manufacturing paradigm has to be set up to make these individualized medicines. And right now, many eligible patients are just not able to receive these treatments because of manufacturing constraints. Uh, One quote from Sloan Kettering at the end of last year clearly shows that they can only treat 2 out or 3 out of every 10 patients with these CAR T therapies because they just can't make enough of them. And then the other problem that is faced is that these medicines are by definition heterogeneous because you're taking a mixed population of T cells from a patient and editing them and then putting them back into the patient. You don't have a pure population of cells. And so when you're doing testing of these, if you don't have the right testing considerations, in many cases, the potency test, which accompanies a good manufacturing process and product release, would yield a result that would say that your medicine is out of spec. And of course, it's going to be administered anyways because of these manufacturing constraints. But what that means to the manufacturer of these drugs is that they can't actually get reimbursed for these therapies because they didn't meet GMP processes. So there's this potency QC valley of death for the drug manufacturers that the medicine in often cases still works. It's just that the testing right now said it wouldn't, and then they can't charge for it. And then the third question that is very obvious is that since every patient requires its own drug, every drug needs to be tested for potency. So this means that the whole framework for individualized drug testing of patient-based cell medicines has to be grown up in order to satisfy the demand for these drugs. So despite these really glaring issues and the very high cost of these drugs, just because they're so individualized and manufacturing is much more difficult, these markets are large and they're growing very rapidly. And that's because these drugs really do provide a true cure for cancer. These are cell-based medicines. They're injected into the body. They're your own cells. They recognize the target and eliminate them. And then these cells also grow so that they can maintain a continued durable immune response over time if that cancer ever returns. That's very different than small molecule drugs, which are more or less treating symptoms and hoping to eradicate any of the cells that are resistant, but those cells may come back later. So there's some issues now that and FDA and drug manufacturers are very excited about these therapies because of the high success rates. The question of just how do we understand the mode of action of these drugs and what makes a characteristically good drug has been a difficult question to answer. And both the industry and the regulators are grappling with this question of what sort of measurements are even needed for cell-based medicines. And I think the result is that came out of the recent guidance that FDA put out to the industry last year was that we don't exactly know right now what are the most important metrics to measure, and therefore let's measure a battery of different metrics, a matrix of assays that should be accompanied for commercial lot release. And these, I would like to, in the rest of this talk, go through my view of what some of these matrix of assays are actually asking, and then how we are trying to address these. So what are the measurements needed in a QC release assay? One of the main things that is needed is to test a test for potency. And they specifically call out cell-based potency tests. FDA has called that out in the matrix of assays that should be used. And these typically are T cells that measurements that show whether the T cell, first of all, kills the antigen-presenting cell. And another measure of potency is whether they secrete molecules to recruit other cells either to directly kill the cell through the secretion of molecules or to recruit other cells to mount an immune defense. And I think you just saw a talk earlier in this lab session about the second part, the secretion of molecules. In this talk, I want to focus more on this direct cell killing assay. Secondly, we want these therapies to directly and specifically attack the antigen presenting cell, the target, but will not harm the other cells in the body, which are the non-antigen presenting cells. So there needs to be tests also for specificity of the therapy. The cells obviously are living drugs, so they must be viable, and there will be company obviously tests that ask whether the therapy immediately prior to administration is viable, and there are metrics for that, which are very clear. There are also tests that are needed to just understand, because you are taking cells from a patient and editing them with CAR molecules. We need to know what percentage of the edited cells actually do express the CAR in the right format that can participate in the therapy. And then finally, there are just impurities from a manufacturing process that have to be dealt with, like beads and contaminant cells, which in many cases can lead to resistance against the therapy, as well as biological contaminants like mycoplasma and bacteria that need to be tested to make sure that you won't induce a bacterial infection upon administering the therapy. So all of these together are the package that is needed for a QC release assay, among others that are all specialized for every drug manufacturer and accompany their CMC controls in the clinical trials. So what my belief is that since these medicines are inherently heterogeneous, bulk-based techniques are not going to adequately characterize the heterogeneity of these living medicines. And I think it's an understatement to say that in many cases, the drug that you inject into the body and the drug that maintains that response over time, that population of cells will evolve. And it's usually going to be a minority fraction of the original sample that was edited is the one that is leading to the durable response. So any sort of bulk method for characterizing the heterogeneity is not going to be answering this fundamental question. So in spite of that, there are good reasons to do bulk potency testing, and I just want to review the current state of the art for that. One is just based on whole plate time-lapse imaging, where T cells and cancer cells are put into a dish together, maybe stained differentially with fluorescent colors so that cell counting of the T cell population and the cancer cell population over time can be monitored through time-lapse imaging. And by tracking these different populations, you can get assessments for cell killing. Another similar approach is based on impedance measurements. So here, electric current is being passed through the aqueous media, and the presence of cells will change the impedance of that measurement. So it is an indirect measure of the total population of different cells, adherent cells on a substrate, and how they may change over time, because these electrical measurements are non-invasive and allow for testing of cytotoxicity and proliferation of both target and immune cells. And then there's just the basic flow cytometer, which is capable of counting cells at different time points. So, and you can imagine a typical assay would be you would mix red and green T cell and effector cells together and let them participate in the co-culture killing assay. And then at the beginning time point and at multiple time points later, you can measure population shifts that would tell you the response of the therapy. But all of these techniques are really measuring the average response. They're not getting at the functional heterogeneity or the rare populations of cells that are actually going to be the most potent and that are going to thrive and proliferate and become the dominant response in the clinic. So this is one of the reasons why current QC tools and potency metrics are not very well correlated with clinical responses. So to address this question, many groups have realized the importance of single cell functional phenotyping. And this is purely my view about how I tend to categorize things, but I view the field as being three main groups. One group is technologies based on droplets or particles. So these are either water and oil emulsions that co-encapsulate cells, and sometimes cell co-cultures, or particles that can capture cells and they can be used in diagnostic tools such as flow cytometry. So these are nice tools for getting high throughput, and they're low cost and they're compatible with flow cytometers, which allow them to screen hundreds of thousands or millions of these particles over time. But the problem is that there's no way to correlate the population at time point zero, individual cells within that population, to other time points. So you really only get kind of single time point measurement, which doesn't help help you measure the dynamic heterogeneity that exists in the sample population. The other group, and the ones that we're working on, is our microwells. So the advantages of microwell technology, these consist just generally of plates where you have little tiny wells at the bottom of each well. These wells are designed to be around the same size as the cell. So cells that sediment into these microwells tend to be one or two, and by assembling more than one type of cell into these wells, you can get these co-cultures. So it is a platform that biologists are very familiar with, usually a 96 well plate. And it can, there's no microfluidics, so it's easy to do time-lapse measurements in a microscope and maintain them under an incubator. And it tends to be low cost. One of the downsides of this process is that it's inherently a stochastic assembly process. But in many ways, what I'm going to show you here is that we also view this as a fundamental feature, not a bug, of the system, because it allows you to test all of your controls and then different effector target ratios in the microwells all in one process. So we actually think it's a feature, an advantage. Then the third category is basically microfluidic methods, which have more deterministic control. They can precisely create specific cell populations of one T cell, one cancer cell, and they can also do time-lapse measurements, but these systems tend to be somewhat high cost and not very reliable because they require continuous pumping to maintain these cells over time. So that's why we have focused on microwell technology, because we think it's the most robust and low-cost way that can meet both cost requirements and scale and variety of measurements that are needed for next generation single-cell functional assays. So to give you kind of when we went through this journey, we had to surmount a large number of technological challenges to be able to do these measurements on enough number of cells at enough number of time points. So just to give you a sense of the scale of this problem, consider that if we have a 96 well plate and we're imaging at least 2,000 microwells per well, we are going to take movies of 200,000 microwells in one experiment in hourly intervals. And some of these experiments may last several days, up to 4 days, where you'd have more than 100 time points in 5 channels. So when you think about the scale of this problem, you're actually having to analyze more than 100 million images in each experiment. And so the first question becomes, how do you even acquire that many images all in perfect focus? And I'll talk a little bit about how we did that. So in a sense, and if you think about it in microwells images per second, you have to take 300 microwells per second images of them continuously over many days. So then, assuming that you even are able to capture all these images, the next question is, how do you analyze all them? And ideally, you want to analyze these images faster than you are acquiring these images, so that all the data analysis is done by the time the experiment is over. So that's another challenge that we faced and are in the process of surmounting. These measurements also have to be non-invasive. So we have to pay a lot of close attention to what sort of imaging conditions we're using, and temperature and other sorts of environmental conditions that can perform these image measurements non-invasively to watch the dynamics of these cells over time, similarly as though they were in an incubator and in the perfect conditions. So assuming that we're able to now acquire 200,000 movies, then the next question is, how do we visualize this data? Which movies do we actually look at? And how do we group these and decide which ones are the important events that tell you about the heterogeneity? So we actually faced a lot of challenges, even internally, for validating our own AI models, was to figure out which movies to even look at. And then finally, suppose you are able to do all that and you can decide which are the important events that you want to look at, then retrieving these clones for deeper characterization is a feature that is definitely needed for understanding the combination of phenotypic and genotypic properties of specific functionally vetted T cells. So this is what we worked on over the past 2 years. And I want to give you some examples of how we solved this problem. So the first problem that I'm pretty proud of is our single shot deep autofocusing tool. So focusing takes up a lot of time in imaging systems using gradient-based techniques that takes a number of images and then decides what's the right focal plane. What we've done is to develop a single shot autofocusing tool that takes one image at some arbitrary object focal plane. And from it, it predicts exactly how far out of focus it is and then jumps in the next frame directly to the focal point. So what I'm showing here is that this dot here is an image taken where we know it to be 175 microns out of the focal plane, but the prediction of the AI says it's off only by about 5 microns. So it thought it was 170 instead of 175 microns. It can jump right to the focal point within the depth of field of the objective. So that has dramatically sped up our imaging processes. The other things that we've done, because we have to take 300 microwells per second images, we need very large fields of view cameras with high numerical aperture. And in order to increase the speed of imaging in terms of 4 or 5 fluorescent channels, we need to have multi-camera systems that can acquire 2 different fluorescent signals at the same time. So when we combine all these together, we're able to achieve area scans that allow us to approach hourly imaging of a full 96-well plate in hourly in 5 channels. The next question that we had to solve is, suppose we can actually acquire and crop and stack all of these images to make time movies of each microwell. How do we actually analyze the contents of these microwells for events that are sometimes very difficult even for our own eyes to decide what's happening? So for example, like initially when you have a T cell and cancer cells sedimented into the microwells, and one example is shown here, the T cell immediately latches on to a cancer cell and starts to interrogate it and potentially to perform cytolytic processes. And the question there is, like, can our AI models, computer vision models, actually distinguish 2 cells in close proximity and tell them apart? And then also tell which is the cancer cell, which is the target cell, because we need to understand what is the composition of these co-cultures and how they evolve over time. So we did a lot of work with just testing different strategies for doing cell segmentation. And a classic example is where we stain the T cells in green and the cancer cells in red, and we just basically train instant segmentation models to do cell counting in these microwells, where we test these across either just using the bright field image or adding in 1 channel or 2 channels or just using the fluorescent images. And so we performed a head-to-head characterization to determine what is the best approach for doing cell counting in microwells. And surprisingly, what we found is that the bright field models, though slightly inferior to ones that require fluorescent overlays, performed reasonably well, meaning that in 80% of cases, for the large part, the model is learning more about the morphology of the cell, the shape, how it moves, the granularity of the cell. It's using that to pick out and distinguish one cell from another, more so than using the fluorescent color of the cell. So as an example of what we mean by that, and what we are showing here is a validation metric for these models. So on the x-axis here, we're plotting the number of effector cells per microwell, and on the y-axis, the number of cells that the computer thinks is in a microwell. So anything along this axis is a correct response. The human and the computer agree that there are 3 cells in a microwell, and anything off this axis would be either over-prediction or under-prediction by the computer. So when we're providing those graphs of accuracy, what we're doing is really saying that what percentage of points lie along this diagonal relative to the total number of points. And from that, we can get this 80% accuracy. And this is actually means that if it thinks there's actually 3 cells and the computer predicts there's 4 cells, it's an inaccurate call, even though it's still probably pretty close. So it just shows you the capabilities of these deep learning tools for doing cell detection in complicated systems, where in some cases you see cell killing and then a lot of debris floating around as well. So that was very promising. And based on these results, we started to do analyze statistical distributions of when we're creating these co-cultures, how do they organize themselves in the microwells? So what I'm showing on the left is one well, where there are these a little more than 2,000 microwells that we've taken time series of in this well. And we have plotted them with 3 colors so that we can clearly distinguish the different types of co-cultures. The green diamonds are the effector monocultures. So these are microwells that only have T cells. And the red squares are target monocultures. So these are microwells that have only spherical, looking, K562 cells presenting the CD19 antigen. And then co-cultures are the ones that have the T cell and the cancer cell in the same microwell. So another way of viewing this well map would be a E to T trapping ratio, where on the x-axis we have the targets per microwell, on the y-axis we have the number of effector cells per microwell. So what we have in this system is actually all of the controls are built into the experiment, because we naturally are going to get the target monoculture controls and the effector monoculture controls. But we also get the entire effector target ratios all randomly assembled in the experiment, so that we're able to actually do these sorts of potency tests that they're doing in bulk dishes all within one well. So cells, which are in green, and cancer cells in red. Oops. I forgot that. Audio. So we have T cells, which are in green, and cancer cells, which are in red. And then over time, we see that the target cells grow if they're not in the presence of the effectors. And if they are, in these 3 examples here, where we started off with exactly 1 T cell, 1 cancer cell, these are cases where the T cell killed the cancer cell and then grew afterwards. Because if you, what are the hallmarks of a good living medicine? These are medicines where the T cell will kill the target, but then also expand to create more copies of itself to continue to mount that immune response in vivo. So this is a kind of an example of what sort of data that we get. And in the previous graph, what I showed you was the ability to assemble all the different effector target ratios in one well. So this is an example of an experiment where we did 6 wells. So each of these, we conformed all of those statistical analyses. The blue wells are non-target cells. So these are wells where we added K562 that did not have CD19, but they are co-cultured with the effector cells. And what we show here is that the growth rate of the non-targets is not affected by the presence of T cells. The purple bars here are the same scenario, but where we added K562 cells that had CD19 antigen. So these are the target cells, And what you see over different effector and target ratios is that the greater the number of effector cells proportionally to the target cells, the more killing that you get. So this shows you specificity of the response. Again, noting that we are being able to perform these experiments all in one well. And the replicates are just biological replicates. So that's why we did 6 wells here, because we had 3 wells where we did the exact same experiment, looking for repeatability, compared to 3 wells where we did the controls. So what should be you should start to learn from these when you do these experiments is that there is a heterogeneity in responses of the effector cell population towards the target cell. So one of the better ways of visualizing this is to plot the target division rate, which is what the x-axis is here, in terms of the number of doublings per day. So the 1.0 would be 1 doubling per day. 1.50 would be a fast-growing cell doubling 1.5 times per day. And on the y-axis, we have the effector cell division rate. So this is a 2-dimensional growth rate plot where each microwell represents, each data point represents 1 microwell. And so wells, and what I'm showing here are specifically co-cultures versus wells that are monocultures in green or red. So obviously for T-cell monocultures, you don't have any cells, target cells, so they don't have any growth rates. The scatter here is just to help us visualize the data points. Likewise, for the target cells, there are no T cells, so you don't have any effector cell growth rates. But these are ones where we have both T cells and cancer cells on the left together, and you see 2 different distributions. One is where the target cells don't grow, but the T cells do. So these are the potent cells, and they lie predominantly along the y-axis. And then you see the cells that are the microwells along the x-axis, where the T cells did not grow, but the target cells did grow. So these are T cells that did not kill the target, and the target just proliferated like normally. And so one of the things that we needed to do at Celldom internally was just to validate that these data that we see on a plot like this are actually what the movies look like in experiments. Developed a data visualization tool that allowed us to click on individual data points to open up movies. So what I clicked on here was a microwell that had a high growth rate along the CAR T axis. And so we see that this cell is proliferating over time. Then we can use these filter tools to only plot the effector cell population. And here I'm showing a target cell that is growing at a normal rate, dividing about once per day, which is what you see here. So this allows us to then look at specific examples of co-cultures to observe what are the rare events that are happening in these samples, like which are the ones that should be candidates for further characterization. So here's an example where I click on one along the y-axis, where there is a T cell and a cancer cell initially at T0, but over time we see that the T cell both kills the cancer cell and then grows out. So once we have these tools to both acquire all these images and analyze them in real time, and then present and help us select which ones would require deeper characterization, the next logical step is to develop a cell picker that can then do genomic testing on it. Audio has been unmuted. So here's an example where we're coming in with a cell picker to retrieve the cell population within a specific microwell. And so what we're doing at Celldom is pairing this liquid handling / cell picking capability with high resolution real-time microscopy that allows us to find hits, pull them out, and then either develop cell lines from them or test their gene expression characteristics or learn more about what makes these cells more potent killers. So this is one of our key applications that we're working on. Another of the interesting areas that we've spent significant time on, also using the same microwell technology, is the measurement of drug resistance in cancer. And so in this experiment, instead of forming co-cultures, we randomly deposited a cancer cell line into microwells and then measured the growth rate of these cells in different concentrations of background drug. And a lot of the reason why therapies fail from small molecule therapies is that there are some of these rare drug resistant cells which still proliferate despite being exposed to the therapy. And then over time, these turn into a drug resistant tumor. So being able to identify which of these cells within a population are drug resistant using these functional characterization tools paired with a cell picker to retrieve these cells to do whole genome sequencing or transcriptional analysis of these cells would really provide a potential game changer, again, for a precision medicine scenario of testing patient samples across an array of different drugs before they ever even receive the drug therapy and start driving that resistance response. So just as a high resolution movie of this, you can take a look here, and what you'll see, this was taken at every 30 minutes over the course of 7 days. So this is the random initial seeding distribution, and for the first 3 or 4 days, you really cannot see a big difference between any of the microwell populations. But over time, and in the center of the screen, you start to see one of these cells, the drug-resistant cells, start to grow out of the sample. And in the background of all of the other cells, which are drug sensitive and either perhaps don't die, but at least don't grow. So the ability to do these high throughput measurements and measure cellular trajectories over time in very large throughput, and then analyze these images also in real time, allows us at the end of the experiment to decide which clones to pick, in this case, a drug-resistant clone that we can retrieve for deeper genomic characterization. So that's been our general strategy at Celldom, is we recognize that the biology field, specifically for functional cell biology at single cell resolution, needs a complete platform to solve these big problems. And so what we have developed here are fit for purpose live cell imagers that include liquid handling and cell picking. So that's a mock-up on the left of what our instrument looks like. We just produced this instrument, and we now have it available for early adopters. This instrument is a 4-channel fluorescent imager with an onboard environmental control system that controls gas, humidity, and CO2, and has a cell picker that can retrieve individual cells for downstream characterization. We've also developed a consumable that's in a 96-well format, and all the results that we've shown here today are produced off of this consumable, where at the bottom of each well, there's an array of microwells. So the reason we chose a 96-well format is because we could do multiplexing of different conditions to test many different patient samples on a plate, or many drugs on a plate, or many of the aspects of a QC potency assay all within one plate. And then we spent a lot of time on integrating software to make sure that these measurements can be performed in real time and allow the user to actually find these live cells, which have an expiration date. So we have to pick them quickly at the end of the experiment, otherwise they will not survive. And so this strategy really requires an integrated approach that pairs the biology with the instrumentation and hardware, when the software all have to work together to do functional characterizations and lead selection for cell biology. So with that said, we are looking for collaborations, scientists and pharma companies that have interesting target applications. We think that this tool is particularly useful for T-cell testings, immunological co-cultures, and even antibody discovery using cell-based presentations. We think that this platform also will have a lot of interesting applications in biologics discovery for identifying TCR sequences using these cell killing assays, or antibody sequences that either are effective or suppressing a response in a cell-based target. And also cell line development problems where the next generation of producer cell lines are needed to be developed. Often cases, with for precision medicine scenario where each of these will only serve one patient and enough number of cells, viruses need to be produced. This platform can be a very interesting approach to high throughput cell line development. And then finally, precision medicine testing. And I presented an example of drug resistance testing, but there are many scenarios where you can envision this as a clinical diagnostic that could track patient responses over time by quantifying the outgrowth of resistance in a therapy and minimal residual disease. So with that, I would love to hear from the audience and address any questions and have a discussion. So thank you for your time.
Well, thank you, Dr. Yellen, for that informative presentation. We will now start the live Q&A portion. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have a few questions. Our first question from an audience member: what time resolution do you need to capture the dynamics of these cellular processes?
Yeah, that's a great question. And it goes right to the core of why you need a microscopy platform to be flexible in terms of the scale of the problem of interest, as well as how fast these processes occur. So, an example, I'll give a couple extremes here. In the example of a T cell potency assay, we need to capture an initial time point right away. So that before the T cells actually killed the cancer cell. And so therefore we need the entire plate to be imaged in ideally less than an hour, perhaps even a half hour. And then to watch killing dynamics, you need one time scale, hourly or even several hours is fine. To watch growth rates, on the other hand, you need days. And so there you perhaps don't need a measurement every hour. You may need a measurement every 6 hours. And then there, in some cases, throughput of the experiment is more important. So we kind of are at the happy medium here, that we can image as fast as we want in an experiment, and we can just choose not to image faster than that. But that's why we stuck with a 5x objective and a 5-channel imager that only requires 9 images per well, so that we can image stage positions at about once per second, which is about as fast as imagers can run. Later on, we would work with customers that have higher throughput applications, specifically for drug screening, where they only need to image once every 6 or 12 hours. We can integrate plate handlers into the system to get through higher number of wells, but fewer time points per well.
Thank you so much. Now, why are bulk measurements inadequate? And this is a two-part question. Why are measurements inadequate, and why do you need single cell resolution?
Yeah, this is the kind of a related question, I think. So the reason what I think commonly happens in these therapies is that, specifically for cell-based therapies, is that a small minority of the edited cells are actually producing the majority of response in a patient. And the way they can learn about this is by taking serial biopsies of a patient over time and performing single cell sequencing. And this is what studies have shown, both in clinical trials and other academic studies, is that relatively few of the clones will emerge over time, and there's a relatively small number of these clones compared with the clonal diversity of the original population. So it's clear then that only a small fraction of the edited cells are the ones that are doing the killing and then are also proliferating in the body. So therefore, in order to characterize clinical responses, we need a tool that can actually measure the the same sort of questions initially. Specifically, growth weight, we think, is one of the main ones that needs to be measured that would actually reflect what's happening in the clinic. So that's why you just would never see that in a single cell measurement, a bulk measurement of like what is the distribution of growth rates of a cell, as well as which ones kill. And then when we add in other measurements, which ones secrete cytokines, which ones do these other things, all that together may provide a better set of biomarker tools that would be indicative of clinical effectiveness.
Thank you so much, Dr. Yellen. It looks like we have one more question coming in. Which measurements will have the highest correlation with clinical responses?
Yeah, excuse me. It's my own personal this is my own personal view here. So I think that what makes a living medicine potent is that it's one that not only is able to kill, but is actually able to expand. Because what you really want is the good killers to be persistent. And in the field of CAR T, when we talk about persistence, people usually describe it as the ability of one T cell to kill many more cancer cells. But it doesn't take into effect anything that that T cell also can grow and make copies of itself, and each one of these can now kill. So taking advantage of this exponential growth process is not something that I have seen a lot of work looking at right now, because these tools just were not possible to really measure single cell growth rates. But in terms of clinical responses, what I'm expecting is that if a prospective or retrospective clinical trial was conducted, where at the initial time point the clonal diversity of an edited sample was performed or measured, and then over time, as that was tracked, uh, we may find evidence in this initial assay that's based on just growth rate and killing and a few other properties, a relatively few number of these, which may have the highest correlation with the clinical response. So we're gearing up to do these studies with partners that have access to clinical samples and want to develop better indicators for their CAR T therapies.
Thank you so much. And Dr. Yellen, I want to thank you for your presentation today. Would you like to provide any closing remarks for our audience before we go?
Yeah, I don't really have much else to say. That we're just at the beginning stretches of this journey in cell and gene therapy, and we don't really know what are the correct measurements to be made on single cells currently. And I think that there's a lot of good work by many groups on measuring secreted molecules, measuring transcriptional processes of live cells using luciferase assays, and measuring cell killing processes. That the ultimate goal of this field is to have a combine all of these measurements into one data set that can correlate every single cell and all of their functional properties as well as their genomic properties. And this also has to be affordable because in CAR T field, the manufacturing costs are just astronomical right now, and this is limiting the ability to actually apply these therapies. So tools that are in the single cell space not only need to provide more diversity in measurements, they also have to be conducted in a high throughput, but also very economic format, so that this can be deployed at a wider scale and lower the cost and provide greater accessibility to these breakthrough therapies. So that's I think we're at the beginning of this journey, and it'll be really interesting to see what happens. But my final also closing remark is that we have these instruments that we're in San Carlos in California, and we would love to perform collaborations with any local parties, or also we can also supply instruments that could be delivered to you for experiments in your facility. So please get in touch with me at Yellen at Celldom.com.
Well, Dr. Yellen, I want to thank you again for important research and for your time today. Before we go, I want to thank our audience for joining us and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed for 2 years until March 22nd, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.